Hey everyone, welcome back to the channel. In today's high yield session, we are diving deep into one of the most important and the most complicated topic in pharmacology, that is pharmacodynamics. In simple terms, science that explains how a tiny pill can cause a massive effects on the body. So, pharmacodynamics is nothing but mechanism of a drug action. By the end of this guide, you will understand how drug interacts with the body with the real life examples and a simplified breakdown. Let's see now the definition of pharmacodynamics. Pharmacodynamics is the science of what drug do to the body. It answers how drug works, where it works, what is needed for a response, how different drug interacts with each other. Key principle in the pharmacodynamics is drugs never create a new functions, they only modify the existing biological process. Now let's see the principles of drug action. Drugs don't create a new functions in the body, they only change how fast or how strongly something already happens. This can lead to useful effects but sometimes also the side effects. So let's break down the 5 main types of a drug action. First one is a stimulation. In here, the drug increases the activity in certain cells or an organs. Example, adrenaline makes the heartbeat faster, whereas the pilocarpine, it acts as an cholinergic and boosts the salivary production. If the stimulation is too strong, it can later causes the opposite, like tiredness or shutting down. Example, very high doses of an CNS stimulants like a picrotoxin causes the seizures and then coma. So coming to the next drug action is depression. The drug slow down or reduces the activity of a specific cells. Example, barbiturates slow down the brain activity in the CNS, and the quinidine reduces the heart activity. And the coming to the other example, that is omeprazole, that decreases the stomach acid. Some drugs can stimulate one part of the body and depress the another. Example, acetylcholine speeds up the gut movements but slows the heart rate. And coming to the third drug action, it is irritation. The drug causes a strong or a harsh reaction, especially in a less specialized tissues like skin or a lining cells. Example, the corrosive chemicals can damage the tissue, leading to the swelling, burns, or even the cell death. This kind of an action is often non-specific and can be harmful. Coming to the next type of a drug action, that is replacement. The drug replaces something that the body is lacking. Example, insulin for diabetes mellitus. When the body doesn't provide the enough insulin, if you take the external sources of an insulin. And the next example is an levodopa for the Parkinson disease to replace the dopamine. And also we take the iron supplement to treat the anemia. So these are especially helpful in deficiency conditions. So coming to the last one, last type of a drug action, that is cytotoxic action. The drug kills cells like bacteria or cancer cells while trying not to harm the healthy ones. Example, penicillin kills the bacteria. And this coming to the second example, that is chemotherapy drugs kills the cancer cells, and Zurovidin, an antiviral, fights against the HIV virus. These drugs are targeted to destroy bad cells without hurting you too much. So to sum it up, while drugs don't create the new functions in the body, but they play a powerful role by enhancing or reducing or replacing or irritating or destroying the specific cellular activities, forming the foundation for how they produce the therapeutic or harmful effects. Now, that's we understand the basic types of a drug action. Let's move on to how these actions are carried out under the mechanism of drug action. So far, we have seen what kind of overall functional impact does a drug have. When we talk about the mechanism of drug action, we are focusing on what the drug does at the microscopic level, the specific molecular targets it binds or it interferes with. These targets includes receptors, enzymes, ion channels, and the transporters. And sometimes the drug works simply through the physical or a chemical properties. So let's break it down one by one. First one is receptor binding. The drugs acts like a keys and the receptors are like a locks. When the drug bind to the receptors, like example GPCR receptors, G protein coupled receptors or nuclear receptors, it either activates the receptors like turning on the switch, or it blocks like tapping the switch off. Next, coming to the important terms in the pharmacology is agonist or antagonist. Agonist means turns the signal on. Antagonist means block the signal, that is turning it off. Example, adrenaline binds to beta 1 receptors in the heart, so and thereby increasing the heart rate. When coming to the other example, loratadine blocks the H1 histamine receptors and decreases the allergic symptoms like rash, itching, etc. Then coming to the next one is an ion channel modulation. Ion channel acts like gates for the charged particles like sodium or a calcium. Some drugs work by opening or blocking these channels, which directly affect the nerve impulse or a muscle contraction. Example, lidocaine blocks the voltage-gated sodium channels to prevent the brain transmission. When coming to the other examples under the ion channel modulation is amlodipine that blocks the calcium channels, so it relaxes the blood vessels, thereby reducing the blood pressure. This is essentially important in treating the arrhythmias, seizures, or hypertension. So coming to the next other mechanism of action through which the drug act is enzyme interaction. Enzymes are the body chemical catalysts. The drug can either block or boost them. When drug inhibit the enzyme, they will stop the enzymes from producing a certain chemicals. Example, captopril inhibit E enzyme, that is angiotensin converting enzyme. So thereby stop forming then angiotensin 2, thereby reducing the blood pressure. Coming to the next other example with the allopurinol. Allopurinol inhibits the xanthine oxidase, so that crucial, which is the crucial enzyme in the production of the uric acid, thereby it reduces the uric acid secretion, treating the gout. This mechanism is precise and commonly used in cardiovascular, metabolic and neurological drugs. So coming to the other example, that is through the transporter inhibition. The transporters or the proteins, they carry the substances across the cell membranes. The drug can block these to increase or decrease the amount of substances in the cell or a bloodstream. Example includes fluoxetine blocks the serotonin reuptake transporters and thereby increasing the serotonin, which treats the depression. And the other example for this transporter inhibition is furosemide antidiuretic blocks the sodium, potassium or 2Cl transporters, thereby promoting the urination, so treating the edema and the hypertension. This is crucial in the drugs affecting the nervous system, kidney and gastrointestinal tract. So coming to the next one, that is physical, that acts as a physical or a chemical actions. Some drugs don't need to bind to anything. They act through their physical or a chemical nature. They might neutralize the acid, bind the toxins or draw water through the osmosis. Now let's see the few of the examples. First one is an example here is a bulk laxatives like esophageal husk. It works by increasing the stool mass. Coming to the next example, that is osmotic agents like a mannitol and magnesium sulfates draws water into the intestine or kidney tubules. And coming to the next example, that is an antacid, it neutralizes the stomach directly. So thereby it's like a chemical reaction. Acid and the base form the neutral substances. Then coming back to the other example is chelating agents like EDTA or dimercaprol bind and remove the toxic metals from the body. These mechanisms are simple but powerful, the kind of like using the physics and chemistry to nudge the body in the right direction. What is the big takeaway from here? Each drug has specific mechanism. It targets a particular pathway or a structure in the body. Understanding this helps us predict its effects and possible side effects and how it might interact with the other drugs. It's not just what the drug does, it's how it does is that matters. Up next, we'll look at different types of receptors and how drugs interact with them in a real world clinical scenario. Stay tuned. Now imagine this, your body is full of tiny locks hidden on the cells everywhere, and guess what? The drugs are like tiny keys. When the right key, that means the drug, finds the right lock, that means the receptor, the magic happens. But wait, it's not just about fitting in. The keys has to turn to make something happen. So that's where two big ideas came in. One is affinity and the second one is intrinsic activity. So first we look at an affinity. Affinity is how well the drug bind to the receptors. Think of like a magnet, whether if it is a strong or a weak. So then next, coming to the intrinsic activity. Intrinsic activity is what happens after binding. Does the key turn? Does it trigger an action inside the cell or no? So here the journey is in simple steps, that is drug bind to the receptors with some affinity. If after binding they do some an intrinsic activity, thereby providing the pharmacological response. And while this happens, a hidden network called the signal transduction mechanism carries the message deep into the inside of the cell to produce the effects like an muscle movements, hormone release, or a pain relief. Now, based upon the intrinsic activity, the drug fall into 4 different types. One is an agonist. That means these are the full heroes. They bind tight and they trigger the maximum response. Example, adrenaline rushes your heart. Coming to the next one type is a partial agonist. Almost these are guys. They bind and activate but only halfway. Example, buprenorphine in pain control. Coming to the next one is an inverse agonist. Those are like rebels. They bind and do the opposite, flipping the action in reverse, negative activity. Example, beta-carbolines causing anxiety. So now let's see what are beta-carbolines and how they work. Beta-carbolines are the group of the chemical compounds that bind to the GABA A receptors in the brain. But instead of calming the brain, like an GABA normally does, some beta-carbolines actually reduces the GABA activity. Normally, GABA A receptors, when they are activated, causes the inhibition, thereby calming down the brain activity, reducing the anxiety symptoms and promote the relaxation. But when coming to the beta-carbolines, they act as an inverse agonist at an GABA A receptors. This means they don't just block the GABA, they actually do the opposite. That is like decreasing the inhibitory signals, increasing the brain excitability, and triggering the anxiety, restlessness, and even seizures at the high doses. So when you hear beta-carbolines causes the anxiety, it is because they reverse the calming effects of GABA, making the brain more excitable and the anxious. And coming to the final one, that is an antagonist. The blockers, they fit perfectly but refuses to open the door. No action themselves, but they stop others. Example, propranolol calming a racing heart. Final takeaways here are: drugs are a keys, affinity locks them in, intrinsic activity turns the locks, and that's how action begins. Alright, now let's dive into the world of receptors, the amazing communication hubs of the body. There are 4 main classes, each with their own unique superpower. Let's meet them one by one. The first one is ionic receptors. These are the fastest guns in the town. Like they are ligand-gated ion channels, meaning as soon as the drug binds, the channel pops open, letting the ions rush in or out. These are the fastest reactions like a nerve impulse and the muscle movements happening through these guys. Examples are GABA A receptors and serotonin receptors and other nicotinic receptors. And coming to the next one, that is an enzyme receptors. These are the master controllers, also known as tyrosine kinase receptors. When a drug binds, it triggers the enzyme cascade inside the cell, like turning on a whole machine factory. They control the growth, metabolism, and healing process. Example includes insulin, a growth hormone, prolactin, and cytokinins. Think of flipping a big switch that powers an entire control room. Coming to that is GPCRs, that is G protein coupled receptors, the most versatile and flexible of all the receptors. These work through G proteins inside the cell, triggering the secondary messengers like CAMP or calcium or directly affecting the ion channels. They can amplify signals and the fine-tune body responses, handling everything from the heart rate to the mood to the digestion. Example, adrenaline on the beta receptors increases the CAMP, fastens the heartbeat. When coming to the adrenaline on the alpha receptors, it acts by increasing the calcium, thereby blood vessel thinning. So think of GPCRs as in smart switchboards routing the messages across the body. So coming to the final one, that is intracellular receptors. These are the slow but powerful editors. These receptors are inside the cell and only lipid soluble drugs can reach them. After binding, they move to the DNA and change the gene transcription, rewriting the instructions of how the cell behaves. Example, corticosteroids, sex hormones like an estrogen and the testosterone, thyroid hormones like a T3 and a T4, vitamin A and a D, and PPAR activators. Think of them as an secret agents changing the blueprint inside the control room. So here the takeaway points are: ionotropic receptors are the fast gates, enzymatic receptors are the enzyme switches, GPCRs are the versatile signal routers, and the intracellular DNA editors. Each receptor type is an hero working differently: fast, slow, direct, or deep inside. And now let's deep dive in. Let's zoom into the fastest communicators in the body. These are the amazing ionotropic receptors. These guys are lightning. They act as an ligand-gated ion channels, meaning the moment a drug or a chemical binds, the gate instantly swings open or closed. Ions rushes across the cell membranes, triggering the immediate effects like nerve signals firing or muscle contraction. Key features of these ionotropic receptors are: these are the fast-acting, directly open or close the ion channels upon binding, and the rapid onset of action. Effect starts within a milliseconds and the shortest duration, quick to start and quick to fade. Now let's meet some real life ionotropic receptor heroes and the drugs that works on them. First one is an GABA A receptors. This is the calming shield. The drug here is diazepines like diazepam. Bind to the GABA A receptors, enhances the GABA action, thereby boosts the chloride ion influx, due to which neurons get hyperpolarized and prevent the overexcitation. So what is the result of this? It results in calmness, sedation, and anxiety relief. To the other examples under the ionotropic receptors is NMDA receptors. These are like an excitation blockers. The drugs like ketamine or memantine binds to the NMDA receptors, blocks the glutamate, which is an excitatory neurotransmitter, thereby reducing the influx of calcium ions, so thereby inhibiting the excitatory signals and protecting the neurons, leading to anesthesia or neuroprotection used in the surgeries or dementia. And coming to the next example under the ionotropic receptors are AMPA receptor. Those are seizure stoppers. The drugs like sodium valproate. Once the sodium valproate binds to the AMPA receptors, it stops the sodium influx, thereby preventing the depolarization, thereby reducing the neuronal firing, the result of which seizure will be controlled, protecting the brain from the overfiring. Coming to the next example under the ionotropic receptors. Now let's see a bit deeper into the NMDA and AMPA receptors. In the heart of the brain's memory center, the hippocampus, two powerful gatekeepers of the communication stand side by side. One is the AMPA receptor and another is the NMDA receptor. When a whisper of an electrical stimulation reaches a neuron terminal, glutamate, the brain master messenger, is released. It binds to both receptors, but with a gentle touch only. AMPA awakens, sodium ions flows, and a slight spark of an light stirs in the postsynaptic neuron. At the resting stage, the NMDA receptors remain silent, guarded by the vigilant magnesium ion, blocking its channel, allowing barely a trickle of the current to pass. But when the stimulation intensifies and the neuron depolarizes, the magnesium barrier is lifted, calcium rushes in, and with it the orchestra of the chain begins. The calcium binds to the colmodulin, awakening a CAMP kinase, a molecular maestro. A CAMP kinase strikes two critical nodes. It phosphorylates AMPA receptors, boosting their ability to carry the sodium and amplifying the signal. And it mobilizes the hidden AMPA receptors, bringing them to the surface, ready to receive and strengthen the communication. Behind the scenes, Calcium also sends a retrograde whisper back into the presynaptic terminal, a nitric oxide signal, urging it to release even more neurotransmitter. So, what does it result in? A synapse that once responded weakly now roars with the power. This enhanced communication, this remarkable strengthening is called the long-term potentiation or LTP. LTP is the cellular symphony that underlies the learning, memory, and the very essence of the experience itself. Ionotropic receptors is nicotinic M receptors. These are muscle relaxers. When the drugs like a succinylcholine bind to these receptors, they open the sodium channels, causing the sustained depolarization, thereby resulting in muscle desensitization and the relaxation. This is the main reason before performing the surgeries, doctor give the anticholinergics for the muscle relaxation. Coming to the next type of ionotropic receptor is nicotinic N receptors. Those are alertness boosters. The drugs like nicotine binds to the nicotinic receptors and receptors, stimulates the autonomic ganglia, thereby activating the sympathetic system and producing the principal neurotransmitters like noradrenaline, which may result in increased alertness, high blood pressure, and CNS stimulation. Coming to the last example under the ionotropic receptors is serotonin receptors. These are anti-nausea defenders. The drugs like ondansetron blocks 5-HT3 receptors, where it prevents the serotonin from overacting, thereby inhibiting the vagus nerve and thereby stopping the nausea and vomiting, especially during the chemotherapy or a surgery. Quick summary here is each ionotropic receptors controls the rapid body responses, and these drugs either boost or block them for specific medical benefits. Now let's see the second type of receptors which are classified under this signal transduction mechanism is the master controllers of the cell. Nothing but enzyme receptors, also called as tyrosine kinase receptors. These receptors are not about the speed, they are about building power step by step. When these tyrosinase kinase receptors activated, they start the phosphorylation cascades, which means adding the phosphate group like adding a power switches inside the cell. They have a slow to moderate onset, not instant but powerful and lasting. Receptors activation triggers enzyme action inside the cell, changing how the cell grows, divides, and survives. Think of tyrosine kinase receptors as slow starting engines that power up a massive cellular factories inside your cell. Now before we dive into the pathways, let's quickly meet some important players. First one is a growth hormone, or a GH, is a powerful hormone made by the pituitary gland. It's like the body's natural growth booster, helps us grow taller, build muscles, and the repair tissues. The other term is JAK2 or Janus kinase 2 is an enzyme that acts like a switch inside a cell. When activated, it adds a little phosphate tags to the protein, starting the powerful signal chain. And coming to the next term is STAT proteins, short for signal transducers and activators of transcription. These are the special messengers. Once activated, they rush into the cell nucleus and turn the specific genes that controls the growth and the protein production. Next up is the prolactin. It stimulates the milk production after childbirth. Prolactin uses the same JAK-STAT pathway to activate the genes needed for the lactation. Coming to the next one is we have the cytokinins, the tiny messenger proteins of our immune system. They help immune cells communicate during the infection, inflammation, or injury, keep us protected and healthy. And tying it all together is the JAK-STAT pathway, a super fast communication routes that connects what's happening outside the cell to changes inside the nucleus, controlling the vital progress like growth, milk production, and immunity. Alright, with these key players introduced, let's now see how they team up in an action starting with the insulin receptors. Insulin bind to the receptor, activated the tyrosine kinase domain, which leads to phosphorylation of an IRS proteins, leading to the two main pathways, that is phosphoinositide 3 kinase protein kinase B pathway and MABK pathway. MABK pathway is nothing but mitogen activated protein kinase pathways. Both are major signaling routes activated after IRS phosphorylation, crucial for controlling the cell growth, metabolism, survival, and the proliferation, thereby increasing the glucose uptake and glucogen synthesis, thereby leading to the lower blood glucose. So coming to the next example is growth hormone receptors. When the growth hormone bind to the receptors, it activates the JAK kinase, Janus kinase domain, which phosphorylates to the STAT. STAT enters into the nucleus, thereby stimulating the gene transcription, thereby promoting the tissue growth and the protein synthesis. Coming to the next other receptor is prolactin. Prolactin binds to the receptors, activate the JAK-STAT pathway. STATs enter into the nucleus, thereby stimulating the transcription of milk protein genes, thereby finally initiating the lactation. So coming to the other receptor under the enzymatic receptors is cytokinin receptors. Examples are interleukins. Cytokinin bind to the receptors, activate the JAK-STAT signaling. STAT regulates the immune gene expression, which in turn promotes the inflammation or a immune modulation that supports the immune responses. So to wrap this up, tyrosine kinase receptors are like a smart control hubs, taking a message from the hormones and the immune molecules and turning it into powerful lasting changes inside the cell. So let's walk through how G protein coupled receptors, the master communicators that activate signals inside this cell. Now let's see the structure of an GPCR and how they are activated. These are the large group of receptors found on the cell membranes linked to the enzyme or a channel through the G proteins. They help control many processes in the body. Coming to the structure of an GPCR, GPCRs have a 7 membranes spanning alpha helical segments with the loops on the extracellular and the intracellular sides. The agonist binding site is on the outer side, while the G protein binds on the inside. So let's see how GPCR works. GCPR activation. When the ligand binds to the GCPR, it stimulates the receptor. This causes alpha subunit to swap GDP for the GTP. Now beta gamma subunit separated from the alpha. Now both alpha, GTP and beta are the free and activated. Second step is signal activation. In this step, activated alpha subunits can now stimulate the CAMP production, increases the calcium levels, or directly influence the ion channels like ionotropic receptors. Beta gamma subunit can also modulate ion channels or other pathways. At this stage, cellular action begins. Coming to the next step is deactivation. After doing its job, the GTP bound to the alpha subunit is slowly hydrolyzed back into GDP by GTPase activity, leading to alpha subunits binds GDP again for forming a stable complex. So after coming to the deactivation, then it'll be coming to the reassociation. Alpha GDP reassociates with the beta gamma subunits, returning to the original stable inactive form, ready for the next signal. So now let's break down the different G protein alpha subtypes, each one like a special command unit inside the cell. First one is the GS, the stimulator. GS stimulates the adrenal cyclase, which leads to the increased CAMP levels inside the cell, resulting in more energy signals, activation of pKa, and many downstream effects. Now, coming to the next other type of an G alpha receptor is alpha I. This is an inhibitor. GI inhibit adenyl cyclase. This lead to decreased CMP production, as a result of which there is a slowdown of a cellular activities important for the signals that calms or suppress the system. Coming to the other subtype of alpha G is alpha O, the calcium gatekeeper. The alpha O inhibits the calcium channels, resulting in which reduce the calcium influx into the cell, thereby controlling the nerves and the muscle excitability. Coming to the next other subunit of alpha receptor is Q. This is the main important one. So it works by activation of an power cascades. So what happens in this exactly is alpha GQ activate phospholipase C beta, which generates two powerful secondary messengers. One is a IP3 and second one is a DAG. So IP3 here increases the intracalcium levels, boosts the contraction and secretions. So when coming to the other DAG, that activates PKC, This triggers powerful cell response. GS is activated, it increases the CAMP levels. If GI is activated, CAMP levels go down. If GO is activated, it closes the calcium gates. If GQ is activated, it will blast open the calcium and the PKC signals. Alright, let's jump into the another incredible molecular adventure, that is the GQ pathway. The first a ligand shows up and bind to the GCPR, then to the alpha adrenergic or muscarinic receptors. Then the GPCR gets activated and it passes the baton to the GQ protein. The GQ alpha subunit ditches the GDP and grabs a GTP, powering up for the action. Next, GQ charges ahead and activates the phospholipase C beta. Phospholipase C beta does something epic. It hydrolyzes a key membrane lipid called PIP2, and from this reaction, two mighty second messengers are born. First one is an IP3, the soluble messenger, and the next one is DAG. This is the membrane-bound messenger. Now, watch the magic unfold. IP3 races through the cytoplasm and binds it to its receptors on the endoplasmic reticulum, and that triggers a flood of calcium ions rushing into the cytoplasm. Meanwhile, DAG stays anchored in the membrane, but here is the twist. DAG and the calcium ions team up to activate the protein kinase C. Protein kinase then goes to work phosphorylating the proteins and sparkling all kinds of cellular responses like contraction, secretion, or a growth. And that's not all. Calcium itself is superstar. It's a third messenger, activating a players like calmodulin, the calcium sensing vis, plus calcium dependent enzymes that drive vital process like muscle contraction and neurotransmitter release. The fun fact here is calcium release isn't just a flood. It can come in the waves or oscillations, fine-tuning the signals into super specific. The key example here this pathway shines is alpha-1 adrenergic receptors. Think blood vessel contraction, and vasopressin V1 receptors that acts on a V1 receptors, and M1 and M2 muscarinic receptors, these big players in the glands and the smooth muscles. And just like that, the GQ pathways power the precision signal across your body. Now buckle up, because this time the action is even faster. It all starts with the GPCR activation, but this time instead of a long cascade, we get instant action. The G protein splits apart, and guess what? The beta gamma subunit and sometimes the alpha subunits dive straight into the action, modulating the ion channels directly. Here, how it plays out. It opens the potassium channels, which make the cells hyperpolarize, meaning they become more negative inside, a braking signal, often powered by GI or GO proteins. This also inhibits the calcium channels. This reduces the neurotransmitter release, another classic move of the GI and GQ. Open channels, on the flip side, GS proteins can actually open the calcium channels, promoting muscle contraction or a secretion. And the best part is, this response is blazing fast, happening in a milliseconds to seconds. Real world examples include the M2 muscarinic receptors act via GI or GO, opens the potassium channels to slow down the heart rate. So, and coming to the next example, opioid receptors like dopamine, D2 receptors and the GABA receptors are heavy hitters using these direct ion channel tricks to dampen the neuronal activity. Alright, now we are going to deeper inside the cell, into the cytoplasmic and even the nucleus itself. Here is the deal. These receptors are not chilling on the cell surface. Nope, they are hidden inside, and they are only activated by the lipid soluble drugs that can sneak the right through the cell membranes. And what's their mission? They regulate the gene expression, flipping the genes on or off, like controlling the master blueprint of the cell. Now, fair warning, these signals are the slowest to start, but when they kick in, their effects are powerful and long-lasting, like setting up a whole new operating system inside the cell. Two main styles of action here is, one is cytoplasmic receptors. These drugs enters into the cell, bind to the receptor in the cytoplasm, then drug-receptor complex marches into the nucleus. Inside the nucleus, it binds to the DNA, alters the transcription and changes the proteins the cell makes, driven long-term changes. Example here is corticosteroids like a glucocorticoids and mineralocorticoids. Coming to the other example, under this cellular receptors is nuclear receptor. These are the drug goes straight into the nucleus, bind directly into the receptors, then grabs onto the DNA and modifies the transcription with sustained powerful effects. Example are PPAR receptors, which are key for the metabolism in the body, and the sex hormones like estrogen and the testosterone. And the other example is vitamin A and vitamin D and the thyroid hormones T3 and T4. In short of this is the drug bind to the receptors, it hits the DNA, alter the transcription, leading to the formation. Stay healthy, stay cheesy with Pharmacy Made Easy. Follow for more.